problem is that, I don't know, it's very hard to figure out.
Especially when you want to simultaneously with small cells.
Now we have two.
Can everybody hear me? Probably, right? Okay, good. First, I would like to thank the organizers, Christina and Roy, for giving us the possibility to present our data. And I would like to talk about a more or less biological problem, about F-actin dynamics that directs velocity of cell migration. Because Margaret gave a really nice introduction yesterday, I just want to talk briefly about the cell migration itself. So this process is required for embryogenesis, tissue repair and wound healing, as well as immune responses. But it is also responsible for tumor formation and metastasis, as well as for some brain defects. Cell migration is a four-step process that includes the protrusion that usually occurs at the leading edge of a migrating cell, and the formation of new adhesions that occur also at the leading edge, and then the contraction of cell movement or movement of the cell body, and finally the dissolution or de-adhesion that occurs at the rear of the back of the cell. So this is an example of a migrating keratinocyte, and you can really see that it forms protrusion at the leading edge, whereas it's retracting at the back. Here you can see actually the retraction fiber. This is, well, it's not so good to see probably. This is in a large region of a cell that was transiently transfected with cerevisiae, and you can see actually besides membrane ruffling, the protrusion as well as the retraction. Besides actin polymerization that is required for cell migration, Rho GTPases play also an essential role in this, during this process. And there are primarily three major GTPases, CDC42, Rho A, and RAG1, that are involved in the formation of specific actin-containing structures. So CDC42 is responsible for the formation of filopodia, whereas Rho A is responsible for formation of stress fibers and focal adhesions, and RAG1 is involved in the formation of lamellar protein. All these Rho GTPases cycle between an active or a GTP-bound state where it can bind to effectors, or they can stay in inactive or GTP-bound state. For our experiments, we used the human oral keratinocytes. This is an immortalized cell line, and the first, or the image on top shows you an immunostain for actin in red and tubular in green, and you can see that this is really a polarized cell with a leading edge where you have filopodia and membrane ruffles present, and at the back you have the action retraction fiber. At the bottom there are you can see a migrating cell visualized with DIC microscopy. Here's a little set of two dividing cells visualized by video microscopy. To study cell migration, today there are actually three ways, three common ways. The first and the classical one is the cell culture, where you have a cell culture vessel, roll your cells, and then monitor the process of cell migration for a certain time period. Then you have that wound closure or stretch assays that you can use to study cell migration. For this, you're using usually a tip, a pipette tip, produce a gap in the middle of a subculture dish and follow the gap closure over time. And then, I never used this actually, but obviously there were some labs that use fluorescent beads, and they followed these beads by fluorescence microscopy, and they were able to track those cells. The next slide shows actually one of these classical scratch assays that we have performed. And here you can see, I hope it's working. So this is for a period of 12 hours. And I will put it again. So here you have the gap, and you can see that the cells or single cells start to migrate into this gap, and over time that this gap is really closed up with cells that are moving into this gap.
How big is that gap?
It's about 200 nanometer, I think. Oh, I'm sorry, yes, yeah. I'm sorry. Good. Because scratch assays are not the best solution in our opinion, or it's not really comfortable to use this, we decided to perform sort of long-term live cell imaging to study cell migration. And luckily, the microscope manufacturers made some toys available. So here we have, and we have tested actually both systems. So one is from Nikon, and this is a so-called biostation. And the other system was designed by Olympus, and it's called the incubator microscope. I just want to talk briefly, or mention briefly, that this system is or contains phase contrast optics, and there's space for only a single 35 millimeter dish, which is sort of limiting. Whereas the incubator microscope from Olympus provides a rotating disc inside the incubator, and there is space for up to 8 35 millimeter dishes, which allows you to do a lot of experiments simultaneously. And in addition, this system contains DLC optics, which allows you better imaging of the cell migration itself. So the next step is, when we've collected the data with one of these systems, then we transferred them into a Metamorph for further motion analysis. So what we have used is that track objects tool, which allows you to place rectangular regions in the area of interest, and over time you can follow the tracking process. So this would be here at the beginning, and this is where the tracking process is right now. And finally, you're ending up with a whole bunch of data that allows you to characterize the movement of individual cells or cell groups. So this, for instance, you can get data about position, velocity, as well as distances. In addition, we performed chymographic analysis to get further information about leading edge activities. So here we have a DIC image of a keratinocyte. We placed a couple of lines in the regions of interest, and you can create those chymographs or time distance plots, which allows you to follow the movement of the leading edge, and you can clearly distinguish between protrusion and retraction activity, as well as you can see that the structures that are moving. So in this case here, we have a lamellipodium that is protruding and retracting, and here you can see a filopodium that is actually protruding or retracting. With this in mind, we have created sort of motility cell migration assay where we can monitor cell groups, not only single cells, cell groups. And this is a movie that covers actually 30 minutes, a period of 30 minutes. And these are, let's say, control cells that were cultured in 35 millimeter dishes. And you can see that you have protrusion and retraction activity, that the cells are sort of connected and circling randomly around. When you analyze this movement with the Metamorph software, you're able to create this migration path, and you can really see that these cells have a non-synchronous or randomly movement. And if you do these chymographic analysis, can see that the protrusion velocity or the instantaneous velocity ranges from about 1 to 1.5 microns. So this is what is pretty straightforward. So you can collect not only data from single cells, but only from cell groups. And the next step we were asking, so how can we alter the cell migration? What can we do to get further information about changes? So and this is when we got in touch with microbiologists. And ladies and gentlemen, I can present now the second key player in our migration assay. And this is Candida albicans that we are adding actually to our cultures. Candida albicans is, as stated here, a human pathogen, and to make it straight, it can actually kill individuals under special circumstances. This pathogenic fungus exists in 3 isoforms. So typical, you have it stays in the yeast form, like Saccharomyces cerevisiae. It can stay in an intermediate state and produce this so-called pseudohyphae, or really true hyphae, which represent a multi-nucleate filament. So when we added those fungus to our cultures, you can see that the migration of those human keratinocytes is changing dramatically. The first thing I want to point out is that you can clearly see that the fungus is growing, so it forms really nice hyphae, and that the cell migration, or the cell sheet is migrating more synchronous and more directed in one way. We don't know whether this is chemotaxis or not, but obviously there's something going on with these cell cultures. When we analyzed these cells further, you can see indeed that these cells move as a sheet, synchronous, and it's more directed for some reason. So analyzing or using kymograph for further analysis, we were able to see that the instantaneous velocity increased dramatically. So now we're reaching up to 4 microns per minute for individual cells. Next, we plotted actually, or we created a graph where we plotted these instantaneous velocities and compared instantaneous velocities of untreated cultures versus cultures that were treated with C. albicans for 3 and 6 hours. And we were able to see that the untreated cultures and even the 3-hour instantaneous velocities for the 3-hour time interval showed a normal distribution, whereas at the 6-hour time interval we were able to pick up a bimodal distribution with actually 2 major peaks, one at around 1.5 micron, and the second one is around 2.5 micron. When we compared the migration rate of these cells that were moving under different conditions, so we can show, or we could show, that untreated cultures move at an average of 1 micron per minute, whereas cultures that were treated with C. albicans for 6 hours, they showed an increase of about 2. What's the time?
Another 3 minutes.
So the next thing we did, we looked more closely to the actin filament system itself, because obviously there are some changes going on that are triggered by the presence of this fungus. So we fixed and stained cells with rhodamine phalloidine at defined time intervals. So when you look at the untreated cultures, you primarily, or you can see that those cells exhibited primarily philopodia. At 3-hour time interval, we were able to see primarily stress fibers and focal adhesions, and at the later time interval, these cells exhibited lamellipodia, extensive lamellipodia. In control experiments, we used Saccharomyces cerevisiae that were co-cultured with these oral keratinocytes. And as you can see, there's not a big difference between untreated cultures and those that were treated with Saccharomyces. And finally, we were interested in whether the actin dynamics is really affected. So we've transiently transfected these cell lines with wired F-actin, and in this image you can see a single cell in an untreated cultures, and I think it's really nicely the protrusion and retraction activity of Philopodia visible, whereas in the presence of Candida albicans, you can see clearly that actin stress fibers and focal adhesions are present, and that the protrusion and retraction cycle is affected as well. So when we co-culture these cells with Candida for 6 hours, you can see a really nice lamellipodium and I would say an increased protrusion and retraction activity. It's actually the same play rate, so these are 10 frames per second. So, and finally, this is our control again, the culture that was treated with Saccharomyces cerevisiae, and you can see that these cells behave similar in terms of actin dynamics as untreated cultures. So, from this one we can conclude that C. albicans, pathogenic fungus, can alter the actin filament dynamics and can increase the cell migration of human oral keratinocytes at early time intervals. We did not observe actin remodeling when Saccharomyces cerevisiae was added to cultures of human keratinocytes, and we can make the statement that C. albicans probably activate the ROC signaling pathway. Finally, I would like to show a working model. We have more evidence, but the time is not enough. This diagram shows the sort of the sequential changes in the actin cytoskeleton caused by C. albicans. So it is well known, and we have shown that this fungus can secrete virulence factors into the medium and cause actually changes in the actin cytoskeleton. So we are starting with Philopodia and microspikes in untreated cultures, but at later time intervals, we see that cells exhibited focal adhesions and stress fibers, as well as lamellipodium and membrane ruffles at the 6-hour time. So finally, I would like to thank some of our collaborators. This is work done by Christiane Rollenhagen, a postdoc in Paula Sundström's lab at Dartmouth Medical School. And this is our former technician, Mary Young. She has developed she made it possible to transfect those cells, since it's a little tricky because you have to keep them at a really low confluent level, and you can't use electroporation in one of these classical stuff that is available. Thank you.
Do you see the same results if you use fungus conditioned media and just add that without the actual pathogens?
We see this effect. It's a little bit delayed. So obviously, you need the presence of the fungus as well. So and I can tell you that these fungus or the fungi secreting SAPs, these are aspartyl proteases that are responsible for these changes. But it is not known what the receptor is on the cell. So if someone can figure that out, that's
Any other questions?
Well, let's thank the speaker again.